1 or PowerPoint 1 Part B. Again, there are the 5 different types of microbes. Please take a peek at your lecture outline if you don't remember them. There are viruses, there are protozoans, helminths, fungi, and again, bacteria. Bacteria is the main focus in our course. So for fungi, there are many different things people think of. It's yeast as well as mold. When you were a young child in a science class, you might have taken a piece of white Wonder Bread, added a little water, and put it into a sandwich bag, and then you grew your own little mold on there. So with fungi, for each of them, I've written it out in bulleted form to help you remember it. And again, please take a look at your outline for some of this extra information. We want to know what type of organism they are. Most importantly, here I'll write this out for you. So what type of organism is it? Is it eukaryotic or prokaryotic, so single or multi-celled? How they obtain their nutrients, and any other specific information that we will cover. So first and foremost, we'll start with fungi. These guys are eukaryotic, which means they are multi-celled, but there are in fact fungi that are unicellular. So the two main types are mold and yeast. How do they obtain their nutrients? They are decomposers. So I'm just going to write nutrients, so when you study and review this, you'll remember. They are decomposers because if you go hiking and you see a big tree that has fallen over, you'll see that it's rotting and there's all types of fungi growing on it. That is the fungi nourishing itself. Fungi is used to provide antibiotics, such as penicillin, that we will talk about later, and they're also used widely in the food industry. So fungi that are pathogenic, which means disease-causing, are dimorphic. Di means two. That means that they are found in more than one shape or size. Mushrooms also fall under fungi. You've probably heard you just can't eat any type of mushroom that sprouts. You need to know if they are the edible kind or in fact pathogenic. Next, we'll look at protists. Many of these are actually waterborne. This slide shows a couple of illnesses. So gastroenteritis can be caused by a protozoan infection. It can cause a sleeping sickness, among other things. Now, the information for protists is this group of microbes are unicellular, which means they are single-celled. Underneath fall protozoa as well as algae. How they obtain their nutrients, I'll just write nutrients for you here, is through absorption as well as photosynthesis. They have the ability to photosynthesize because some of them have a cell wall, and a cell wall is required for an organism to use photosynthesis as a means to get its nutrients. And again, some of the human diseases that are associated with protists from the previous slide and this one are listed for you. Next, we have helminths, which deserve a little bit of an extra explanation. Helminths are traditionally incredibly large in their full form. If somebody has a tapeworm from eating undercooked or contaminated pork, for example, it could be meters long, and there's 3 feet and a meter, so incredibly big. And we are in a microbiology class where most things we discuss are naked. To the naked eye, you are unable to see them without some type of microscope. So you might wonder why helminths are involved in a microbiology course. And the answer for that question is because they have different life cycles. And the life cycles in the beginning consist of eggs and cysts, similar to when you think of lice. Those are in fact microscopic. So we do mention helminths, but there is absolutely no focus on it. The focus is purely always going to be on bacteria. So about helminths, again, they are eukaryotic. They do not have a cell wall. So therefore, if you think back to protozoans and protists that we've spoken about momentarily before, and bacteria, they do have a cell wall, so they get their nutrients. One of the ways is through photosynthesis. In this case, the nutrients are either absorbed or ingested. Photosynthesis is not a source of nutrients for helminths. And again, why do we cover them here? Because they have microscopic life cycles in the beginning when they have the unicellular cysts, eggs, that become larvae, and then at that point, again, they are eukaryotic and incredibly large, such as a tapeworm in a human's gastrointestinal tract. Next, we have viruses. We don't spend a lot of time on it here in the beginning because viruses consists, or our entire Unit 5 and Module 5 consists of viruses. If you just take a look at the pictures on top, you see every shape and every size. This looks like a little alien ship landing somewhere. Viruses are very unique. They are not alive. They are what's known as obligate intracellular parasites, meaning they use the host's systems and processes to replicate themselves. They can't just live by themselves sitting on a table without a host and using their machinery. Later in the class, we're going to talk about viroids and prions, which are something completely different. But for now, a little information on viruses. They have a core of nucleic acids, meaning they can either be DNA or RNA-based viruses. And those viruses have a protein coat on them to actually help protect them. They are obligate parasites, and I would also say obligate intracellular parasites. They are classified by a few different things. First, morphology, which is the same as their shape. Second, through the genetic material, is it DNA or RNA at the core? And then certain biological properties. So how do they infect? Is it a respiratory illness? Is it a digestive illness? What type of virus it is? And again, prions, which are actually protein substances, and viroids will be discussed later in another unit. And now here's bacteria, which is the most important in our class because, again, this is microbiology. A lot of people don't realize this, but bacteria is found absolutely everywhere and anywhere. You ingest it all day long. You put it on your bodies. You put it in your hair. You put it in your mouth. So a couple different things. If something feels slimy, it essentially has bacteria in it. We're going to learn about the glycocalyx later, which is what you are feeling in the slimy. So any lotion that you're putting on, hair conditioner, toothpaste, even something as simple as Vaseline, it contains something called xanthan gum, which is related to and from bacteria. Another example is if you buy, for example, Dannon Active. It's a yogurt that says it has an active substance of bacteria and it has lactobacillus growing in it. This is a probiotic to keep your bacterial counts in your GI tract at a healthy level. So here's the information of bacteria, and I need for you to focus on this when exam time comes. They are unicellular, which are single-celled, and they do in fact have a cell wall. Because remember when we went through the lecture outline, I mentioned that the cell wall composition is the first most important way to determine the type of bacteria and to actually identify it. So how do they get their nutrients? One of two ways. Make sure you remember everything on this slide, please. One is they absorb the nutrients, and the second way is they are photosynthesizers. The next way to classify bacteria is based on their shape, and there are three different kinds. Bacillus or bacilli are rod-shaped, like Tic Tacs. Cocci are actually round, and spiral kind of look two different ways, but you can think of a slinky, that it's sort of rounded that way. So bacteria is used very heavily in the food industry, whether it's cheese, sauerkraut, wine, beer, all over the place. And bacteria is known to cause many human illnesses. A few examples are cholera, tuberculosis, which is TB for short, and the plague. I want to show you this picture, which is a little bit early, since we don't talk about antibiotics and chemotherapeutic agents till later. But I do want to share something. So this is a Petri plate that you are looking at. These streaky lines is the growth of a bacteria called Staphylococcus, or most likely Staph aureus. So within the growth, you see this white, flowery-looking substance. This is penicillium. So penicillin is actually fungi-based, and most of you are more than aware that this is a very popular antibiotic. So as this bacteria was growing and penicillin was put onto this plate, notice this clearing area that you see right here. This is known as a zone of inhibition or zone of inhibited growth. This basically shows that penicillin is effective to stop the growth of Staphylococcus types of bacteria. This will make a little bit more sense once we start talking about streaking plates for the lab portion of the class, but just understand that this is how we test for medicinal uses, what type of antibiotic is effective against which type of bacterial growth. And lastly, I want to look at this slide for a second. This is what microbes do or where microbes are found. When it comes to exam time, expect to know a couple or a few examples. You don't have